I'm pleased to introduce today Eric Moody from Middlebury College. Eric attended the University of Wisconsin-Madison for his BS in zoology and biological aspects of conservation. Um, he also worked at the University of Wisconsin Trout Lake Station for a year and attended Arizona State University for his MS and PhD in biology. He also worked at Iowa State University as a USDA NIFA postdoctoral fellow before moving to Middlebury College, where he's been an assistant professor in the biology department since 2019. Eric's research focuses on using functional traits, particularly stoichiometric traits, to understand how populations and communities respond to and affect changes to aquatic ecosystems resulting from expanding human populations. So thanks for being here, Eric.
Okay, thanks, Stephanie and Bobby and everybody for the introductions, and everyone out there in the audience for coming to see this presentation. Let's get it started here. Right, hopefully everybody can see the screen now. So I'm going to be talking about where and when stoichiometry matters in aquatic ecosystems, and keeping with the theme of this seminar, we'll be using a lot of data from NEON sites to elucidate some answers to those questions. In opening this up, I'd like to think about an aquatic ecosystem that matters a lot to me. I don't know if folks recognize this. It's not a NEON site, unfortunately, but this is fish slough in the Owens River watershed in California, near the Sierra Nevada. And it's a story that I like to tell to start out my seminars. I'm on maybe sort of a PR tour to promote this story because it's really inspired my career and my trajectory in aquatic science, especially in challenging times when it feels like our work doesn't matter. And so I wanted to share it with you all. So at this location holds one of the last remaining populations of a species called the Owens pupfish. It's a species that was sort of stranded in these isolated desert environments as sea levels receded several thousand years ago. And they became isolated in this one river basin, the Owens River Basin, where they formed a separate species that we now call the Owens pupfish. And it was not far from here where the last known population of Owens pupfish was thought to have gone extinct in the 1960s. But an intrepid fish biologist named Phil Peaster and found a remaining population, much to his surprise, and realized they were still alive, but they were in trouble. And he was monitoring this population pupfish when he noticed the water level was starting to recede. The lone habitat that was known where the species still existed was beginning to dry up. And he took it upon himself to carry all the fish that he could catch in 2 5-gallon buckets across the desert. This was in the middle of the night when he had no flashlight even with him. And the sole survivors of Owen's pupfish, the species still exists today, thanks to his efforts, because that spring dried up later that next day. And they only survived because he carried them to this place, Fish Slough here. And so he wrote this article about it, calling them the species in a bucket. And I find it really inspirational that one person working in a remote place can have such a big difference just by being so attentive to the ecosystem. This quote in particular from his article really strikes me, where he says, "For a few frightening moments, there was only myself standing between life and extinction." And I may not be able to say the same about myself, but I hope that the research that I'm going to present today at least can convince you that some of the things that we're doing matter, and some of the things that NEON's doing really matter for advancing our understanding of aquatic ecosystems. So the species, the fish continued to thrive in this spring where they were transplanted to, but they didn't do so with some bumps along the road. And one thing that we've really learned from this experience and from others is that if we just focus our management on single populations of single species, we are not going to be very successful. There's increasing numbers of studies like this one from Min et al., this one from Rodinger et al., certainly plenty more, that show us that taking a broader view, an ecosystem-based management approach perhaps, is going to best serve us in managing our resources, whether those are fish populations or anything else that we might value about the natural world around us. And so I take that view and I use a particular lens. Some of you may be familiar with it, but some of you may not be, called ecological stoichiometry. And if you know stoichiometry from an intro chemistry class that you took, and that's your only familiarity with that term, then you're still on the right track, because what we're talking about is balancing chemical reactions. Here we're thinking about the chemical reactions of life. So we talk about ecological stoichiometry. We're talking about the balance of multiple chemical elements and energy in ecological systems. So if we think about our pupfish here, not quite to scale, but you can imagine there's a bunch of these fish swimming around in the spring. They need to eat to survive, and they need to get enough food. So the food quantity, how much energy they're getting is important to them, but also the quality of that food is going to be important to them. What is it actually made of? What chemical elements are present in there relative to the demands of those fish? The fish then is going to take that energy and those nutrients and use them to build its body tissue. So it's going to need phosphorus to make bone and scales. It's going to need nitrogen to make proteins. It's also going to need that energy that's in there to fuel its own metabolic rate and assimilation efficiency of those food resources. However, some of those things can't be used, or they can't be used with 100% efficiency. And so we have things like nutrient excretion, the excretion of unassimilated or unused or perhaps metabolic byproducts of nitrogen and phosphorus, as well as the respiration of CO2 after cellular respiration has produced ATP. And then those nutrients and that energy might go back, and some of it might feed back to primary producers in the system. And so there's a cycle here, and we're interested in studying those balances or those imbalances across these different steps. So if we think about it in maybe less of a fishy view, but more of a sort of a diagram of the system, we could think about these key nutrients. We think about nitrogen and phosphorus a lot. They tend to be things that can limit primary production. And so the relative availability of those nutrients can in turn influence things like the community of phytoplankton or algae that are growing in a system, as well as things like the rates of production. In turn, those things then might influence higher level consumers like fishes and benthic invertebrates. So we might think about populations of species that we're interested in managing, or communities that we're interested in protecting. But those populations, through things like excretion, can turn around and also influence the relative supply of those nutrients at an ecosystem scale. And so can the composition of those communities. So which species are present, how many of them are present. These might play an important role in nutrient cycling at an ecosystem scale, as well as other ecosystem rates. And we're really interested in these stoichiometric traits, as we call them, these traits like how much carbon, nitrogen, and phosphorus an organism needs, and how much of those things it might excrete back into the environment, at what rate it's doing those things, because those traits can directly link responses to environmental change, these things that we might think of as the classical phenotype, like life history strategies does an organism use? What defensive structures does it have? How good of a competitor is it under different conditions? To things like the effects that organism might have on an ecosystem. So if organisms that have these sort of R-selected life histories that grow really rapidly and produce lots of offspring, also perhaps require more of a particular nutrient like phosphorus, then we might expect that shifts in the environment that select for certain traits of an organism might then have consequences for the cycling or the pools of nutrients within an ecosystem. So there might be a lot of consequences for these changes to this elemental phenotype or the stoichiometric traits of the consumers and the population. And this was work that was done by Rana El-Sabawi and colleagues. I was not a part of, but really has inspired and dovetailed really nicely with things that we're thinking about. So this is a paper that we have in review led by our postdoc, Ben Tumalo, or former postdoc, recently moved on to another position that will hopefully be soon. You can find the preprint copied in the chat, I believe. But we've been thinking about how stoichiometry can really be an important control on various processes across scales and types of ecosystems, because you often see these nonlinear dynamics, these threshold sort of dynamics occurring when stoichiometry is involved. And that's because you might see a shift in nutrient limitation as you cross a threshold from having a certain nitrogen to phosphorus ratio, where you might move suddenly from being nitrogen limited to phosphorus limited. And we've proposed that this can occur in systems and controlling evolutionary processes like adaptive radiations of plant lineages that colonize islands with unique soil properties, to ecosystem rates like decomposition rates of leaves when they enter a stream, to things like nutrient recycling by consumers, to the reassembly of communities after a disturbance. And so all these things, depending on the traits of the organisms involved, could show these sort of nonlinear controls resulting from either their stoichiometry or the stoichiometry of the ecosystem. All right, so that led us to this question: where and when does stoichiometry matter in aquatic ecosystems? Can we start to fill in some of these arrows to think about cases where these stoichiometric traits are really useful in predicting responses to environmental change? And then similarly, we're not full of hubris. We know that stoichiometry doesn't control everything in the world, although some folks in the world of stoichiometry might talk that way sometimes. I am a realist. I know that in some cases stoichiometry may not actually matter so much. And so we're trying to elucidate these types of questions. So, we can think about things like, if we're thinking about population scales, variation in the resource supply stoichiometry might help us do things like understand how to manage a population of threatened and endangered species, like the Owen's pupfish. We've been applying this to study population growth rates of the humpback chub, which is a federally threatened species in the Colorado River system. I won't talk about that today, but happy to answer questions about it. And so we think that these changes in stoichiometry could have potentially important bottom-up effects on populations. They might also affect the functional diversity of communities. And if we want to think about ecosystem function might be altered by some disturbance, we want to understand what kind of functions are being retained, what kind of functions are present, is there functional redundancy, or what sort of functional diversity might result from those changes. At the same time, a lot of other folks have done quite a bit of work on thinking about how these changes to populations and communities might influence things like nutrient limitation, key ecosystem rates like primary production. Some work from folks like Krista Capps and Carla Atkinson have really illuminated a lot of that and influencing things like primary production. Do we want to manage issues like eutrophication in lakes and rivers and coastal areas? Understanding the relative supply of nutrients can be really important for that. So I think this might be familiar to many in the audience, but just in case there's anyone who's not familiar with NEON already that happened to find their way here, we are using data from the National Ecological Observatory Network, in particular the aquatic sites, to try to test these questions about where and when stoichiometry matters. And especially today, we'll be getting into the where, but as NEON continues going forward, we're hoping to get better and better answers on the when component as well. and we'll touch on that towards the end. But we're really fortunate to have been working with Neon over the past 4 or 5 years now, working with data that Neon's been producing, as well as through our assignable asset project that I'll talk about in a moment. Okay, but before we get to that, I'm going to introduce this framework that we've developed that help us think about these stoichiometric traits might change over environmental gradients. And so when on here, this y-axis change in stoichiometric diversity, we're talking about calculating functional diversity metrics using stoichiometric traits. In this case, we'll talk about traits like the foliar carbon and phosphorus concentrations in terrestrial plants is going to be our example. So we could imagine as a basic scenario, if you look at systems that don't vary much in terms of their conditions, we may not expect a strong change in the stoichiometric diversity of those assemblages, and we would say there's relatively low sensitivity to change in these kinds of systems. So here we're color coding these plants based on sort of binned foliar C to P ratios. So blue means high C to P, gray means moderate, and red means low, just as an illustrative purpose here, not actually based on real data. Based loosely on real data, I would say. But where we start to get more interesting is if we start to move in environmental space, how might these things change? So one scenario that we hypothesized is we might see a change in abundance of different species. So the size of these icons sort of depicts their relative abundance within a community. And so we might see what we call equal stoichiometric impact of a disturbance or a change in conditions if all the taxa have relatively similar changes in abundance. Either they decrease in abundance at similar proportions, or they maybe increase in abundance in similar proportions. But if we were to look at the stoichiometric traits of this community, it would be the same as the first one. On the other hand, a more extreme scenario is what we call stoichiometric turnover, where the traits of the organisms present on the other end of the gradient look completely different from those that were on the original end of the gradient or before the disturbance came along. So here now we're moving from a community of plants that had high foliar C to P to one that's dominated by a different suite of species that have low foliar C to P. And we could measure a metric called stoichiometric distance that would allow us to test for this hypothesis. But that might be a little extreme. Maybe something more likely is what we call differential stoichiometric impact, where we see relative abundance changes of some species. So in this scenario, we're seeing species with high foliar C to P tend to be reduced in abundance. Those with more moderate C to P maybe increase in abundance. So we're not seeing totally new traits emerging in the community. We're just seeing the relative abundance of the traits that already existed are changing. But if we looked at the leaves being produced by this community, we would expect them to differ in average in stoichiometry because of those relative abundance changes. And we can measure that using what we call stoichiometric dispersion, which is calculated based on functional dispersion. And then we could also imagine a scenario like stoichiometric gain or loss. So either we're adding species with totally new traits in addition to those that were already present, or we're losing stoichiometrically unique species, but retaining many of the others. And we're still going to see a change in diversity as a result of those changes, especially if those species that are gained or lost were very stoichiometrically unique. So this outlines some potential scenarios, visually at least, for how we might expect a community to change in its stoichiometric traits over some gradient of space or time, whatever gradient you're interested in. So we started to think about what gradients should we look at. And I, having worked in the arid southwestern US and northern Mexico. During my doctoral work, I became really interested in these very sort of temporally dynamic systems that occur there. And so this is a paper that we published a couple of years ago, led by Thibaut Dautry from the Dry Rivers Research Coordination Network, thinking about human changes to flow regimes might potentially influence the functional diversity of communities and streams. If we think about a stream that's naturally perennial in an environment like this, it might be more stable potentially. We might expect high taxonomic diversity, high functional diversity, a sort of consistent ecosystem functioning if we think about whatever rates we're interested in measuring. If we look at hydrograph for a stream like this, we are in a place with very seasonal precipitation patterns. So you could kind of predict that there's going to be roughly 1, 2, or 3 times of the year where a flood might occur, and there's pretty predictably a dry season as well. But there's always water in the stream. The discharge never reaches zero. On the other hand, a naturally intermittent stream might have different species in it because the environment's very different, but it might still have many of the same properties because it's, the organisms that live there are adapted to the particular flow regime that might occur in a naturally intermittent stream. So again, we might see high diversity functionally and taxonomically, as well as what we call maintained ecosystem functioning, rates of ecosystem function that would be expected for a system like this, even though it may actually dry up at certain times of the year. So, if you look at this hydrograph, you can see there's still peaks and flow at the same times of the year as the naturally perennial stream. But during those dry periods, the stream might dry up consistently. And then we contrasted this with what we call an anthropogenically intermittent stream. So one that maybe has started to dry up because of human influences, as depicted by our lovely center pivot irrigation systems and cities and things like that over here. On the left, we might expect lower taxonomic diversity and lower functional diversity and potentially altered ecosystem functioning, because only organisms with certain traits might be able to live in a place like this, where this is a real hydrograph of a river in Phoenix. You could see the discharge. There's still peaks at some of the same times, but if you were to ask me to predict for you on any given day of the year what the discharge is going to be, I wouldn't want to place a bet on that because it's just all over the place. The city is releasing stormwater, treated wastewater into the river channel at all sorts of various times of the year, to the point where you really can't predict when a flood or drought is going to occur. You could on any given day be totally dry or in flood conditions. And this unpredictability might really have a particularly important role in dictating the functional diversity of the community. So we started looking at this using NEON data when we first started this project, and this was a project led by my undergraduate thesis student, Annie Blaylock, who's now working at the University of Georgia River Basin Center. And she was thinking about resource stoichiometry and hydrology together might have interacting effects on the functional diversity of a community of benthic invertebrates in this case. So if we imagine some traits like size at maturity and development time, we might imagine a community that looks like this with each of these circles representing different species, sort of having relatively even distribution of organisms with different sets of traits. Many different trait states are abundant within the community. And so what we thought might select for a community like this are relatively stable conditions, so predictable flow regimes, someplace that's a lot more like the Owens River, fish slough spring with constant spring flow. And one that has lots of carbon relative to phosphorus and its spatial resources, because we might be constraining the development of particular traits that require high phosphorus that might become dominant in the community. And so we thought these kind of steadier places with relatively high carbon to phosphorus ratios might select for this sort of relatively evenly distributed set of functional traits. On the other hand, you might imagine a community where particular sets of traits dominate, in this case, small things that develop quickly. Yeah, that's what we're trying to depict with these species here that, have these particular sets of traits and these large circles representing relatively high abundance. And if we look at the centroid of the trait space in this community, we'd expect them to be dominated by organisms with particular traits, in this case, ones that are often associated with high investment in phosphorus. So a flashy, unpredictable stream is going to select for rapid growth. Rapid growth requires lots of RNA synthesis to produce all those proteins that you need to grow rapidly. And RNA is a very phosphorus-rich molecule. So we might expect that to select for these P-rich traits, in addition to streams where there is relatively high availability of phosphorus. So low C to P ratios might facilitate these consumers that have higher P demands. So we looked across neon sites and we looked, this is the periphyton carbon to phosphorus ratio on the x-axis here, and the invertebrate density is what we're depicting on the y-axis. And you can see there's a negative trend here. So places that have relatively low phosphorus on the right here, high carbon to phosphorus, tend to have lower density than places with higher phosphorus or low C to P. But there's quite a lot of scatter as well. We found relatively weak effects on any of our functional diversity metrics, though. So periphyton stoichiometry is maybe controlling density, but not functional diversity in the neon weightable streams, at least according to what she found. When we looked at hydrologic variation, we calculated this metric called high flow variation, which is a measure of how predictable high flows are going to be over a time series. And I don't have time to talk about how we calculated that, but I'd be happy to dive into that further if you're interested. But we looked at some of these functional diversity metrics like functional dispersion, and we found that, again, there's a negative relationship. So streams that have less predictable flows had lower functional diversity than ones that were more predictable. But again, there's a lot of variation, a lot of noise in these data. And so we did this using some of these more traditional functional traits like life history traits, functional feeding groups, and things like this. And so this was before we had any good stoichiometry data on these invertebrates. And we thought, well, maybe these relationships would be clarified if we could actually directly measure the stoichiometry of these organisms. And so that's what we decided to try to do. Unfortunately, we got funding from the National Science Foundation to do that. Working with NEON, we've been working to build the STOIC database, which is a database of the elemental chemistry of aquatic organisms, NEON being a very heavy component of that database. And I think the database link, we haven't published it yet. We're working on the data paper right now, but the beta version is available, and you should be able to find it in the chat. So we obviously had a huge team. This was just our STOIC team. It doesn't even include the NEON team. I wish I had a picture of all of you too, because it certainly wouldn't exist without you. But what is the STOIC database? So we have corresponding abundance data along with sometimes stable isotope data, water chemistry data, resource standing stocks, and various other metadata associated with all these entries in our database, focusing on just the benthic invertebrates, which are a relatively small component of the database. We have lots of other organisms as well, but largely thanks to the NEON samples that we've accumulated, we've got data now on over 22 orders, over 100 families, over 200 genera. And that's numbers certainly increased since I even made this slide, since we just had another update about a month ago. So we've got a large diversity of trait data now for benthic invertebrates. including all 34 neon aquatic sites and lots of other sites that we've been working on, trying to fill in some of the gaps where certain areas we didn't have good coverage from neon sites or other land use types that weren't really represented in neon. And we're also scouring the literature and getting contributed sources from others, including sites outside North America, to really bolster the scope of this database. And so one of the fun things about this project is it's a really interdisciplinary team. It's the first grant that I've ever worked on that had an art historian as one of the PIs on it. And so we've had a lot of fun thinking about how to break away from the system that we've been using to think about visualizing these complicated data. Right? Neon is a complex entity with lots of variables that exist over various gradients and things. So trying to understand how can we visualize this complicated multidimensional data. We know there's like NMDS plots that everyone loves to make, but those are hard to interpret. And so we turned to people who maybe had never even seen an NMDS plot to get some inspiration for how we might think about visualizing these data. And one of the ideas that we encountered, this was really thanks to our work with our art historian colleagues, was these Mesoamerican counting devices called Nepo-Waltzinzin. And they originated in various indigenous communities in Central and North America, and were used to keep track of various quantities at the same time. They actually use these as wearable devices. Unfortunately, we haven't been able to make the prototype of that yet, but we can use them instead in a graphical version to keep track of various metrics like the abundance or the density of invertebrates, the stoichiometric distance between samples, the stoichiometric richness of a sample, or the stoichiometric dispersion. And so if we have all these quantities, we could sort of put them all together on a plot that looks kind of like this. And so we translated into this two-dimensional space where we show something like time or an environmental gradient of interest along what we would think of as the x-axis. And then we plot all these different quantities along these sort of strings across the gradient. And so if we think about something on the left, that's sort of like our initial conditions, and then we might look at a disturbance, and then we might carry on forward through the present conditions. And we could represent a community as a bead or a circle that's going to be colored based on that particular value varies from the mean. So we've used standardized z-scores to depict these on a scale of very red, which is that particular time point was much lower than the mean, to blue, dark blue means it's much higher than the mean for that variable. So here we're depicting a community where the abundance doesn't change at all after the disturbance because all the values are at the mean. They're all white, which means they're right around the mean. And we could do this for all these different variables. And if we did this, we'd see in this community, nothing at all was affected by this disturbance. They were all right at the mean. So we might consider this scenario to be the low sensitivity scenario, where no diversity or abundance metrics varied from the time before to after this disturbance. On the other hand, we could start to color in some of these things. And now we're seeing a scenario where stoichiometric dispersion was blue, so higher before the disturbance, and then switched to red, meaning it was lower after the disturbance, which means that that community now is dominated by organisms that have a particular stoichiometry that's different than what it was in the initial conditions. And if we plot the C to P ratio of the community, then we can see again it's changing from blue, so high C to P, to red, which is low. And so this is just one example. I could spend a lot more time going through it, but what I decided I think would be more interesting is if we apply this to the neon data now that we have these elemental data for these aquatic invertebrates. And so one disturbance that I'm sure was challenging for those who were working there at the time, but was fortunate for us as scientists to look at the effects of a disturbance was the impact of Hurricane Maria, the streams in Puerto Rico, because that occurred not too long after NEON got started in 2017. So we looked at these invertebrate communities before and after Hurricane Maria to see how they were impacted by this disturbance, and in particular, how their stoichiometry was affected. And so here we're making a plot, kind of showing the same thing that we were just looking at for the Rio Cupeyes neon site. And you can see these metrics. There's abundance, stoichiometric distance, stoichiometric dispersion, stoichiometric richness, and the carbon to phosphorus ratio of the invertebrate assemblage. In particular, what I want to call your attention to is if you look at stoichiometric dispersion, the pre-hurricane values are all blue, meaning they were higher than the mean of the entire time series. Whereas in the sort of couple of years after the hurricane, they were mostly on the reddish colors, which means they were all below the mean of the whole time series. So stoichiometric dispersion decreased following Hurricane Maria. After that hurricane, the community was dominated by taxa with particular sets of stoichiometric traits. And then if we look down at the bottom, we could kind of get some sense of what that is. The community was more in the sort of low C to P ratios before the hurricane, and then kind of shifted towards being more moderate to higher C to P ratios after the hurricane with some variability. So we looked at like who was there, which taxa were actually driving these changes. So if you like bugs, then you'll be excited, and if not, maybe you'll get to learn a little bit about them. Turns out the biggest driver of change were black flies, which are Samelium. If you don't know black flies, you might know how annoying they can be if you're being bitten by the adults of them. But in streams, they have these really cool fans that they hold out into the current and collect particles from the water column. And so they really like areas with really rapidly flowing water. And they tend to be relatively rich in phosphorus. A C to P ratio of 125 may not sound like it's that rich in phosphorus, but for an insect it is. There's also this particular caddisfly, Leucotrichia. It was never particularly abundant in the samples, but was never identified after Hurricane Maria. And it also has a relatively low C to P ratio. On the other hand, the one taxon that really increased in abundance after the hurricane was Adopsyche. It's a caddisfly. They're predatory. They have a relatively high C to P ratio. You could see it's almost about double that of Samelium. And so we could start to think about what might explain these results. The thing that stood out to us was that Samelium and Leucotrichia are both taxa that really like fast-flowing water, riffle habitats. And looking through some of the neon data and talking with staff in Puerto Rico, it sounded like after the hurricane, those habitats really decreased in abundance in favor of sort of slower moving kind of run habitats in the Rio Cupeyes. And so that's the kind of habitat that Adopsyche really likes. And so this was really a case where it wasn't stoichiometry driving any changes in the system, but rather the physical changes to the system may have caused a shift in the stoichiometry of the consumers. And you may wonder, does this even matter? And that's a question that we're certainly still asking ourselves, but there's at least some hint that it might matter. And so these are the neon's total nitrogen to total phosphorus data before and after the hurricane. What you can see is that before the hurricane, there's maybe more variability, but some instances of relatively high Tn to Tp. So if we had our community that had more black flies, and these are P-rich consumers, they may recycle less P back into the environment, which might be driving up that TN to TP ratio, whereas these atopsyche, being relatively less rich in P, they may recycle more P and potentially contribute to decreasing that TN to TP ratio. There's certainly not to say that this caused any of these changes, and I would not want to bet too much on that, but it's certainly a possibility that's really interesting. Is something that I think we should really explore further, and how these kinds of changes can drive changes in nutrient cycling in systems like this. So the take-home message for this part of the talk, just to highlight that stoichiometric traits can serve as a promising link between these community responses to environmental change and the effects that those changes have on ecosystem function. And we've developed this framework to test these. And we're now interested in exploring this further. And so I want to just, in a few minutes, highlight some of the kind of interesting things we're starting to explore more with some of these NEON data. Because as you can probably appreciate working on this network, there's a huge range of diversity in sites and conditions across NEON that can really get us the opportunity to explore what might drive stoichiometric variation in these invertebrate assemblages across the continent. So we started looking at high flow variation because we had found that it had a pretty strong effect on functional diversity in invertebrate communities. And we really didn't actually find much of a pattern here. So flow predictability across the continental scale doesn't seem to be a particularly strong driver of stoichiometric diversity in these invertebrate communities, which was maybe a little surprising, but certainly interesting to find. We then looked at periphyton stoichiometry, and we thought maybe this might play a role. Sort of as we had already found, total abundance decreases in these streams that are relatively phosphorus poor. So this was similar. We're seeing these kind of red values at these high C to P ratio sites. But interestingly enough, what we found is that the C to P ratio of the invertebrates also decreases at these sites. So this isn't a case of the periphyton. It could be a case of the periphyton constraining the community composition, but one thing that's really stands out to us about these sites is the two that are at the far right end of that gradient are the two sites in Alaska. They seem to have very periphyton C to P and very low consumer C to P. And we think there could be some other like seasonality and light factors that are playing a role in some of these patterns. And Spencer Cruz, who's a PhD student at the University of Wyoming, this project is trying to explore some of this some more with his dissertation. So we looked at, we have all these data, so we decided to look at the body stoichiometry across these groups of invertebrates just to see which groups are actually varying, and we didn't have a great handle from the things we had thought might play a role. Maybe this could lead us in some new interesting directions. And so you can see there certainly is some variation. Some groups, like crustaceans, tend to have very low body carbon to phosphorus ratios relative to insects. And if we look even finer, if you know about benthic invertebrates, you might know about EPT. They're our sort of favorite indices that we use for water quality in places like Vermont, at least where I am. And these are taxa that are very sensitive to variation in water quality. And we notice that these taxa tend to have higher body carbon to phosphorus than most of these other taxa that can be more tolerant of poor water quality. And so we thought, could there potentially be some shifts in the stoichiometry of these communities associated with changes in water quality? The interesting thing about NEON is most of these sites are in places that are relatively minimally human impacted. So we're not looking at sites in a particularly intense area of agriculture or an urban stream. But we can still imagine some of the variation that we might expect to occur to happen naturally over NEON sites. And so one thing that we've been thinking about is variation in specific conductivity. Places like Vermont, urban streams have very high conductivity because of all the de-icings that we use. And so we just looked across neon sites in over this range of conductivity. So low conductivity is kind of like a very wet place, like tropical rainforest stream is going to have low conductivity. High conductivity is likely to occur in spring-fed places with high evaporation rates. And interestingly, what we found is that these streams in high conductivity areas have pretty high stoichiometric dispersion, and the taxa that are there tend to have low body N to P and C to P ratios. And so, that harkens back to this idea that these tolerant taxa, like crustaceans and some of the snails, things like this, that can tolerate the salty conditions, do really well. And we might actually see conductivity as a sort of underappreciated driver of stoichiometric variation across communities. We are starting to explore this over anthropogenic gradients in conductivity in Vermont, but I'm going to skip over that for the sake of time so that I can leave time for questions. But I just want to conclude, one thing that certainly stands out to me having done this work, we have a lot more freshwater invertebrate stoichiometry data than we did 5 years ago. Now, having data from all the neon sites, we're really poised to answer some really interesting questions using neon. We can use these data to link community changes to ecosystem effects, and we're starting to think about ways to do that. We know that unpredictable high flows can drive community change over space and time, but the stoichiometric impacts of those flows are maybe less extreme than we had initially thought they would be. On the other hand, I'm really starting to advocate that thinking about salinity and variation in salinity might be an overlooked variable that might control functional diversity of aquatic invertebrates. And so with that, I'd like to just acknowledge funding from the National Science Foundation, Stephanie Parker, and all the NEON aquatic field staff who contributed to this work, sending us samples, giving us tours of sites over the years. We're immensely grateful for all of you. The Stoich team, a whole lot of people contributed to this, a lot of undergraduate students in my lab and folks at our collaborators' labs as well. I've got my email here if you have questions you want to reach out to me directly. But with that, I'd be happy to take any questions you all have.
Thank you so much, Eric. That was super interesting. I always think it's really interesting to see researchers use several different neon data products to put their story together, and you're using water chemistry and inverts and algae and all sorts of things. So I'm going to start off with a question from Eric Sokol. Do similar stoichiometric responses seen in the macroinvertebrates show up at higher levels in the food web?
We haven't looked at, like, let's say the fish community, and so that would certainly be a good next step. The good news is there already are a lot more fish stoichiometry data because people have been using them as models to study these questions for longer. So we certainly could look at that, but we haven't directly addressed it yet. So stay tuned.
Great. I'm curious, you were talking about the network, and you started off with data from neon sites, and then you said you had an additional 50 data from 50 other sites. Is that another network or just places that all of your collaborators are working? How did you pick up 50 additional sites
Yeah,
worth of
that's a
data?
great question. So we got this project was funded by the EPSCoR Track 2 program, which is funding research collaborations across different EPSCoR states. And one of the things that they had as part of that program was supplemental funding to work with institutions and bring like other states in smaller institutions. And so we got some funding to sort of expand our project a little bit, and particularly thinking about collecting data across land use gradients in streams where some of our collaborators were already working locally in like Alaska and Arkansas and Vermont. And so really the goal with that was to try to kind of fill in some of the gaps that, like, there was unfortunately, was no urban component to neon in the end. And so we really wanted to look at, with urbanization increasingly as a prevalent stressor for streams, how that might potentially play a role across some of these different geographic areas.
Great, thanks. Another question from Sikander. Does the Stoich database have a time series have time series data of abundance and water chemistry in the same locations.
So the database itself contains only the stoichiometry data and some of the associated metadata, but by working with Neon, we know then that like the associated time series of other variables are available for those sites. And then for the other sites, we have a column where we mark that there are other sorts of data like those. Like we have, you'll be able to search it and see data for which we have other abundance data. And so like these other sites that we're describing would, fall into that category. And so we haven't integrated it directly into the database yet, just because there's so much to do already. But certainly there would be ways to then take those data and merge them together.
Great. Some follow-ups. Eric Sokol, is the high C to P ratio a trait that is general to sensitive taxa? Also, does stoic diversity reflect functional diversity?
Yeah, those are good questions. So the first one, I wouldn't say it's universally true that sensitive taxa have high C to P ratios, but a lot of the taxa of invertebrates, especially that are sensitive to water pollution, are sensitive because they have things like tracheal gills that influence, they're affected by pollution, and they tend to have softer exoskeletons, and those things sort of feed into the ultimate body stoichiometry of those organisms. So there's definitely a relationship there that is caused by some of those traits that those organisms have. The second question that you asked, sorry, now I forgot it. Can you repeat it?
Does stoic diversity reflect functional diversity?
Oh, right. So, to some extent, both those things are going to depend on the taxonomic diversity, but in some cases, they're going to differ. And so it really depends on which traits you're using to calculate, let's say, functional diversity, because, you might find things that have, similar types, let's say, life histories that maybe have more rapid growth rates and higher phosphorus concentrations. And so in that, if you're using like those kinds of traits, the stoichiometric diversity is more likely to reflect that. But if you're using traits that are more functional feeding groups or things like that, you can have predators from very different groups that actually have very different stoichiometry. So I think it reflects phylogenetic diversity much more than it does functional diversity. A lot of these traits seem to be phylogenetically conserved, and that's another area that we're working on.
Great. I've got a couple other questions here. One is sort of long. I'm going to boil it down to basically what challenges or insights do you think having standard total nutrient data in combination with scattered types of other data will present to moving ecological stoichiometry forward? So, for example, neon total N and total P data in comparison to historical trends and patterns.
Yeah, it's a challenge because as we found in building this, everyone has different research questions and collects different data or collects data in different ways. And so standardizing it has been challenging, but this highlights why having a network like Neon is really so valuable, because we can make somewhat generalizable conclusions about patterns at such a large scale using a standardized network like Neon. And then, if people are interested in trying to apply those to different scenarios or at a more local context, could then build off of that and test it at a local scale and think about what aspects of those questions that you're most interested in. So, I wouldn't say that anything that we find at a continental scale is necessarily going to be generalizable to, a local scale, like the work that we're doing in Vermont, for example, within a watershed. But it could lead to some insights that, might be worth testing. Um, that, that that's the best that I could say for that.
Great. And last one at the end, you started to talk about impacts of salinity. Have you started to tease that apart, looking at lower and higher salinity sites?
Yeah, so that's what we're working on now in Vermont. We have a gradient of urban streams, or people might laugh when they say urban streams in Vermont because it is a very rural state. But in Burlington, there are some streams that are relatively heavily impacted by impervious surfaces. And one of the main things that we find in those streams is very high salinity, very high chloride concentrations from the de-icing salt that we use in the winter here. So we've started using field data in combination with some experimental data to tease apart how the invertebrate community responds to urbanization and salinity, specifically as a driver of that urban stressor, which is something that one of my students will be talking about at SFS if you're going to be there next week. So we're starting to get there. We've got some funding now from the Lake Champlain Basin Program, which is a local organization that's going to help us really expand on that study. And in a few years, I'll have a better answer for it.
Great,
So
thank you so much.
yeah, thank you very much, Eric, for presenting. Just a reminder, this is our last seminar of the season. If you'd like to nominate yourself or someone else to be a speaker for next year's seminars, Samantha put the link there in the chat. And then also just a reminder to consider applying for the Convergent Summit. Those applications are due at the end of the month. So thank you, everybody, for attending, and thank you, Eric, for presenting.
Thank you.